Welcome to our conversation on sexual reproduction, meiosis, and patterns of inheritance. This is going to be a really interesting series of talks that ends in the kinds of patterns and pathways that lead to evolution and natural selection and provide all of the diversity that nature pulls from. It's very interesting, but it all starts with the process of meiosis and sexual reproduction. Now, there's a lot of different examples of diversity in nature, but usually when we think of a species, we think of something visually that is consistent. That's just a bias from us being human beings that use vision as one of our primary senses, but it's still a good pathway to understanding the kind of diversity that exists genetically in organisms. So this example on the left is a bunch of different frogs. These are all types of poison dart frogs, and you can see there's a ton of different colors. Some of them are red, some of them are blue, some of them have black spots, some of them have shading. Well, it might surprise you to know that all of these frogs are the same species. So there's some mechanism of diversity that's being maintained. There's some way that this species of frog has achieved all of this diversity in its pattern within its population. Here's another example of a single species with a lot of diversity in its visual morphology. Here's a group of snails called the painted snails, and these are from Cuba. But you can see these are all one species of snail, but there's a ton of different colors and patterns, and you might guess just finding one of these snails and comparing it to another that they're different species, but they're actually the same species. So again, we have an example where there's a lot of diversity within the same species, and that diversity is being maintained, and there's some kind of process to allow that diversity to exist. Now, in our species, we also have a lot of diversity. You can see that the way people look changes depending where you're at, and there's just a huge diversity of people. So one question you might want to ask yourself at this point is which stage introduces that kind of variation? You can see here there's the development of a person from an infant to a toddler to a child to an adolescent. Then they're an adult going to school, and they get a job, and then they get older, and finally they pass away. Well, this is kind of the cycle of human life, but which stage actually makes that kind of variety that we just saw in those last few examples? Well, if you're guessing it's somewhere in the beginning, you're right. In this case, we have a toddler that's developing. Well, the actual stage where the variety has been created and this person has been selected from all the different possibilities, that actually happens before the development of the embryo. Before sexual reproduction can occur, we have to make special cells, and these cells carry genetic information, but it's in half. We've halved the genetic information, and there's a very good reason for that, and we have a special name for these cells. We call them gametes. When these gametes meet, an egg is fertilized, and that fertilized egg undergoes the embryonic development that we saw in our last lecture, and that becomes the embryo. But in order to make that fertilized embryo, you need these special cells. They're called gametes, and in our case, they're haploid. Remember, our genome is made up of 23 pairs of chromosomes. Looking at this example, at let's say chromosome 1, we have a chromosome on the left and a chromosome on the right. Well, one of those chromosomes came from the genetic mother, and one of those chromosomes came from the genetic father. What if each parent gave all of their chromosomes? In this case, we're looking at an example where each person has 3 pairs of chromosomes. The genetic mother and the genetic father all have their own sets of chromosomes from their genetic mother and their genetic father. So if they were to just combine chromosomes, they would end up with a tetraploid offspring. That offspring would have double the amount of chromosomes that is normally occurring in the species. This would not be a good scenario, and that individual would not survive. So, what's the solution? We need some mechanism or some way to reduce the number of chromosomes of each parent. So that way, when they're combined, they make a full normal amount of chromosomes. In this case, a pair, one from each parent. If we could do that, then we would have offspring that have pairs of chromosomes instead of sets of 4. We call the number of sets of chromosomes in a cell the ploidy level. When we create gametes like sperm and egg, and remember that these are cells that have reduced the number of chromosomes so that when they combine, they can make a normal amount, those cells are haploid. They have one copy of each chromosome. So diploid means 2 sets of chromosomes. The nomenclature to signify diploid states is 2N, and that means you have 2 sets of each chromosome. Haploid is represented by 1N, and that means we have 1 set of those chromosomes. There are some organisms that have more than 2 sets of chromosomes, and we call those polyploids. They may be triploid, they may be tetraploid, they might have any number of chromosomes. As long as they can reproduce and pass that down, well, that's a stable state for that species. For us, we're diploid. Here's 2 different life cycles to compare so you can start to get an understanding of the kind of diversity that we find in nature as far as ploidy states. So on the left we have the life cycle of Homo sapiens. You can see that after meiosis, after the production of gametes on the far left, we have 2 small cells, an egg cell and a sperm cell. And these are haploid, these are 1N, these have half of the genetic information. They quickly combine through fertilization, and now we have a diploid cell. That cell has all the information that it will need to create the full organism. That one cell becomes the embryo, it becomes the baby, it becomes the toddler, the adolescent, the adult. It undergoes all of that development. So in this case, we would say that the diploid stage is dominant. You and I are diploid. The only stage that we were haploid was when we were separated as an egg and sperm cell. Our species is diploid dominant. On the right is a kind of moss. Well, the mosses are a little different. They spend most of their life stages as haploid. They're 1N. They have half of that information. And then when a sperm and egg combine, at the top of the haploid structure, they create a diploid structure called a sporophyte, and it grows and then it undergoes meiosis and it sends out more haploid spores. Those spores grow as tissues, and those are haploid tissues, and they are the dominant stage in the moss's life cycle. So this is way different than us. But this is an example of how nature doesn't just have 2N dominant life stages. There's also 1N dominant life stages. Where does meiosis occur? Well, in animals, meiosis occurs in 2 places. In males, it occurs in the testes, and the cells that are produced are sperm. These gametes are 1N, sperm have half the genetic information. In females, meiosis occurs in ovaries, and the product are eggs, and eggs have, again, half the genetic information. So when you combine the egg with half the genetic information with the sperm that has half of the genetic information, you get a complete diploid cell, which can then develop into an embryo and then a mature reproductive organism. In the flowering plants, this all occurs within the flower in different structures that we'll explore later in the semester. In the female plant structures, meiosis occurs in the ovary. The gamete is something we call an ovule. Meiosis occurs in male flowering plant structures in the anther, and this produces pollen grains, which is essentially plant sperm. So we should familiarize ourselves with this life cycle, the life cycle of Homo sapiens. This is our life cycle, and it's very easy to relate to. You can see that this cycle is describing the different kinds of cellular reproduction that occur in each stage. And what I always found confusing about these life cycles is that it makes it look like we spend half of our life as sperm and egg. Well, that's not really the human experience. most of our life and most of our experiences are with other organisms that are diploid, 2N. And while we're in our 2N states, as we're growing, as we're developing, we are reproducing our cells through the process of mitosis. And that's what we learned about in our last lecture. So throughout our lives, we're growing and we're developing using mitosis, which just duplicates our genetic information and duplicates the cell structures. It's only when we are preparing to reproduce that we undergo meiosis. Meiosis occurs in specialized parts in our bodies, and it's not something that we're necessarily always focused on, but it's important to remember that meiosis is what is kind of the bedrock of diversity. It is the thing that creates cells that are not exactly like each other. And when those cells combine with other cells from someone else that also aren't exactly like each other, we get this kind of random assortment of features and genes. And that creates the kind of diversity that we see. So while this life cycle appears to be half haploid gametes and half diploid adult organisms, our experience is a little different than that. But you just want to keep in mind that the reason we're emphasizing the stage of egg cells and sperm cells and haploid gametes is because this is a stage where a lot of the diversity gets set. This is where it arises.